So we talk about apoptosis. We call it programmed cell death. It is very similar. Many people, they just consider them as the same process, same term. But for researchers who are doing this kind of research, they actually say that they are two different things, two different processes. But anyway, many people study the programmed cell death or apoptosis because by learning how the cell kill themselves, they hope to figure a way to cure cancer. As I mentioned, cancer is basically uncontrolled, unstopped cell division. So I mentioned that chromatin, the differences between chromatin and chromosome. So here is the structural difference of the chromatin and chromosome. Normally, DNA is a double helix structure. We talked about this early in the semester. They will be packed and wrapped with some kind of histone protein. This purple ball here, this purple ball again, this is a blue ball, purple ball, they are the histone protein. So chromatin, they DNA wrapped around histone protein. So when DNA is wrapped around, wrapping around this purple ball, the histone protein, then they form something called chromatins. But then when chromatin is start to coil, it's like it's very similar to when amino acid form the secondary structure. Remember I told you the secondary structure is like a phone line. So the alpha helix, and then it continues to it has a secondary structure, and then tertiary structure, and then even a quaternary structure, and so on and so forth. And when the chromatin start to coil in different level of the structure, then you will form chromosome. So it looks like this. On the video, you may not be able to see the details of the picture drawn in here. But when you but if you look at the PowerPoint slides, you can see that the chromosome is basically formed by strands of chromatins. So here, this is called one replicated chromosome because it has two chromatins, and in the center we call it centromere. So I hope you still remember the structure of chromosome. The centromere is different from centrosome or centrioles. We'll talk about centrosome and centriole when we go to mitosis. So DNA again is replicated in S phase because S stands for synthesis. And the two chromatids, they are joined at the centromere. So karyotype, karyo again, it means the nucleus. Karyotype, meaning that we classify the chromosome into, we are able to classify chromosome in pairs. So we have 23 pairs of chromosome. Each pair of chromosome, they look very different from each other. They look different from each other in terms of the length and also in terms of the shape. So display of all chromosomes in the somatic cell is called karyotype. So karyotype is easier to do during mitosis because when chromatin is being condensed into chromosome, then we can look at the shape. So different animal, we have different number of chromosomes. For human being, we have 46 chromosomes. On Wednesday lecture, I already told you that it is the reason why also, have I told you already? The reason there's a compatibility of the reproduction is the way how we define species. If the two animals, they are of the same species, then they can reproduce. Not only they can reproduce, their offspring can survive, and not only they can survive, but also the offspring can reproduce. The main reason is that different animals, if animals of different species, most of the time they have different number of chromosomes. When the number of chromosomes does not match, then even if the cell is able to, even if the sperm and egg can be fused together, then the cell will usually stop at the mitosis. Because, well, the inside machinery doesn't match, then mitosis will not occur. But even if they can occur, then the offspring will be very weak, or they may even die during pregnancy. Or let's say even if the offspring can survive, they will not be able to reproduce, just like when horse made with donkey, then the milk that they produce, the milk can live, but the milk cannot reproduce. So we want to say this is an important point because each pair of chromosomes we have, one is from the father and one is from the mother. We call this homolog. We call it homolog, or we call it homologous chromosome. Homologous chromosome meaning that they are a pair. One comes from the father, one comes from the mother. And the last pair, the 23rd, we usually call this a sex chromosome because we use X and Y. As you can see from the picture here, X chromosome is a lot longer than the Y chromosome. Or you can say Y chromosome is so tiny, so small. Because of this reason, when we talk about genetics in the next few lectures, we assume that Y chromosome does not carry any gene. We assume. But nowadays, we discover that in reality, Y chromosome actually carry a little bit gene. But for simplicity of our course, when we talk about genetics in the next few lessons, we assume that Y chromosome is we call it genetically empty. It doesn't carry any genes. So only X chromosome carry genes. So for this X and Y, it is a male. For female, it is X and X. So remember that the chromosome, each pair, one chromosome comes from father, one chromosome comes from mother. So remember this. Now finally, we start the mitosis. So it says that mitosis can divide into 4 main phases: prophase, metaphase, anaphase, and telophase. Yes, you need to memorize all this. Usually for other textbooks, they like to say that it's sort of interphase. So it has 5 major phases. Interphase. Let me see, in Bio 3B, in your Bio 3B lab, you will be required to look at the slides under the microscope, and then you should be able to identify the cell based on what happened. Based on what happened, what do you see in the nucleus? Then theoretically, you should be able to know, you should be able to tell the cell in which phase. So this is in Bio 3B lab. It is kind of difficult if it is the first time you do it, but after you repeatedly practice, then it should be fine. But in our class, since it is a lecture, I'm not going to give you the microscopes and the slides and for you to look at it. But it is okay for me to maybe cut and paste these kind of pictures from the lecture slides into the exam, and then ask you to identify based on what you see on this picture. Tell me the phase. What phase is that? It is legal for me to do this because I'll show you the lecture slides. So the main role of mitosis is to separate the sister chromatids, which we call karyokinesis, and then distribute it in the daughter cell. So you see that when I talk about each phase, karyokinesis occurs first, and then after that cytokinesis occurs at the end. See that cytokinesis occurs at the end. So first of all, the interphase. During interphase, DNA is replicated during S phase before the mitosis occur. And as the picture shows in here, during interphase, they are still in the chromatins. They're still in the chromatins. Well, actually, it is not. It should be prophase here, and I apologize for the mistakes. Prophase. So when after the G2 phase, then the cell goes into the mitosis. The first stage in mitosis is called prophase. During prophase, several things happen. One is that the chromatin condense or pack into chromosomes. So this is the first thing that happens. Chromatin condenses to become chromosomes. The second thing that happens in the prophase is that the nuclear membrane dissolves, or the nuclear envelope dissolves. So the nuclear envelope, I use dissolve, but actually it disappears. So that the chromosomes, they are now exposed to the cytoplasm. And not only the nuclear envelope is break down, but you see that there is. Let's see in the previous slides. So the mitotic spindle. Now we talk about spindle. We see this structure, the two little so-called rectangle. So these are, we call this centrioles. And then if the two centrioles, they are bathed in some kind of gelatinous-like protein. So when centriole and those gel-like protein together, we call it centrosome. So the two things, the two perpendicular rectangles, they are the centriole. And then if we include the surrounding area, we call this a centrosome. And then you have the mitotic spindle coming out from the centrioles. What happened? The third, so in prophase, there are three events. One event, the first event is that the chromatin becomes chromosome. Second event, the nuclear envelope or the nuclear membrane will disappear. The third event is that the mitotic spindle from the centriole, they will attach to the centromere of the chromosome. So remember centromere? Centromere is basically it's the point where the two chromatin connects, and also where the spindle attached to the chromosome. So the spindle will attach to the chromosome at the centromere. So three events occur in the prophase. And then in so it says that the two cytoskeletons are called centrosome. The centrosome, they will move to the opposite end of the cell, and this mitotic spindle, they start to elongate and contract and elongate. They try to position all these centrosomes. So basically, here I just summarize the 3 points that I told you. One is that the chromatin becomes chromosome. Second is that the nuclear envelope disappears. Third is that the mitotic spindle attached to the central mere. And then the centrosome will move to the opposite end of the cell. Now the second stage after prophase we call it metaphase. Metaphase, if you are taking about 3D and look at the microscope, it should be the easiest phase that you can identify, because you see a dark straight line in the middle of the cell. So when you see all the chromosomes line up in they call it metaphase plate or equatorial plate in the middle of the cell, then it is the metaphase. So why would they align in the middle? It is because of the shortening and the elongation of the mitotic spindle of these two centrosomes. So they will adjust the length so that all these chromosomes, they will line up in the middle of the cell. And then the next phase is called anaphase. Anaphase is, what happens during anaphase is that the mitotic spindle, they are being retracted by the centrioles. Centrioles start to collect or shorten the mitotic spindle. When the mitotic spindle is shortened, what will happen? It will pull the two chromatin apart, so that the chromosome will no longer have two chromatin. It will become one chromatin. This is what you see here. When the two chromatin are being pulled apart, then we call it anaphase. In the slides that you see in about 3D, what you would usually see is that you will see the two fingers. It's like you have two bananas or two hands, and the fingers, they are pointing at each other. So in this case, that is anaphase. Telophase. During telophase, so you have several things happen. The spindle no longer connects to the chromosome. The nuclear envelope reforms. So you will see the nuclear envelope now, and most important in telophase, cytokinesis continues. Cytokinesis occurs. So what happens is that if it is an animal cell, then you will have this invagination on the side of the cell membrane. We call it a cleavage furrow. But if it is a plant cell, then you will see a straight line in the middle. We call it cell plate. This cell plate eventually will become a cell wall. And it will continue to connect the two sides into a cell wall. So telophase, when you look at the cell under the microscope, telophase looks like two cells connected to each other. It looks like two cells connected to each other. When you look at it, when you have two cells connected to each other, then it is the telophase. Or you may see one big cell has two nucleus. If you see one big cell has two nucleus, then it is also a telophase. So after telophase, then the cell continues to the cytokinesis. The definition is in the first bullet point. It's the process of dividing a parental cell cytoplasm into two. So what happens is that the actin microfilament, remember actin, those are the cytoskeleton that we talk about in the cell structure and functions. What happens is that they will contract. When this actin filament contract, then it will pull the cell membrane together. It will squeeze the cell so that the cytoplasm will be pinched into two cells. So in this case, we are talking about the animal cell here. because as I mentioned, it is cleavage furrow. How do we spell that? Cleavage furrow. Cleavage furrow. So cell division in plant, they are a little bit different from the mitosis in animal cell. And in my personal opinion, the plant cell the mitosis in plant cell, it is easier to identify each stage, each phase than in animal cell. So what happens is that you can see that, okay, in the interphase, usually you see a black dot, just a black dot. In the prophase, because the chromatin start to condense into chromosome, then you see some, basically you see the nucleus, instead of a black dot, you will see some white spaces in the nucleus. When you see the nucleus that has some white spaces in the nucleus, that is the prophase. The reason is that the chromatin, they condense into chromosome. That's why you have a lot of empty space. And then in the metaphase, you see that all the chromosomes, they line up in the middle. But it doesn't look like a straight line. It looks like, I don't know. You see some, if you pay attention, it is actually a straight line here. And you see some chromosomes, they actually go to the sideway. But in any case, it is remain in the center of the cell. Anaphase, as I told you, that it looks like two hands, they are pointing towards each other, so that the fingers, they are facing each other like this. And then telophase. Telophase is, I told you that it's like two cells, they are connected to each other, or one cell has two nucleus. And as you can, do you see this white line in the middle? The white line in the middle is called cell plate. So cell plate is the precursor of cell wall. And you can see that, okay, anaphase, okay, these chromosomes, they're facing each other like the two hands. And then the formation of cell plate is a characteristic or signature of a plant cell. And then eventually the cell plate will form the cell wall in between the two daughter cells of a plant. Now we finish mitosis. Let's start with meiosis. Meiosis, as we know that, as I repeatedly mentioned, it is for making the gametes. This is the cell division for making sperm and eggs. And each gamete, that means the final daughter cell, they are haploid at the end. So the zygote, as I mentioned before, is the fusion of sperm and eggs together. And after you form the two haploid cells into one diploid cell, then you have the reason we call it a homologous chromosome, or the reason is that one chromosome is from the mother, from the sperm. The other chromosome is from the mother, from the egg. One chromosome is from the father, the sperm. The other chromosome is from the mother, the egg. And then because of that, we call it homologous, or the paternal homolog, or maternal homolog. Together we call it homologous chromosome. Or homologous chromosome. So this is the summary of the meiosis. So at first, the At first, in the eggs and in the sperm and the testes, we have a diploid cell, and then these diploids undergo meiosis, and they will form the sperm and egg, and then after fertilization, they will reform the diploid zygote, and then you will develop using mitosis to grow or develop into the embryo in fetus. To study meiosis, you need to have a very good understanding of mitosis. The reason is that meiosis basically has two stages, meiosis 1 and meiosis 2. And meiosis 2 is the same as mitosis. Yeah. It makes your life a lot easier if you understand mitosis, because meiosis 2 is basically mitosis. So we will focus on meiosis 1. Because meiosis 1 has two significant events. So what happened is that it summarized the purpose of meiosis 1. Meiosis 1 is basically the splitting of the homologous chromosome. The splitting of homologous chromosome, meaning that if you look at this here, in the metaphase 1, it tells you that it is splitting your mother's chromosome and your father's chromosome. So you can assume that in the metaphase 1, it's like your parents are getting a divorce. This is an analogy that I like to use to explain to students. For meiosis 1, or specifically the metaphase 1, metaphase 1 is basically your parents are getting divorced. So that in the telophase, only your parents, they live separately in two different cells. Meiosis 2 is the same as mitosis because it is the splitting of the chromatids. It is splitting of chromatids. So remember the differences. Meiosis 1 is the splitting of the homologous pair chromosome. That means your paternal homologue and the maternal homologue, they will be separated into two different cells. Here, meiosis 2, it will be the same as mitosis. It is basically the splitting of the chromatids. That's why I wrote it here. You need to know the differences between chromatids and chromosomes. Another difference is that meiosis, you will produce 4 daughter cells. So one diploid cell will become 4 haploid cells. But for mitosis, 1 diploid cell will become 2 diploid cells. Yeah, so that's the difference. That's one of the many differences between mitosis and meiosis. Meiosis will form 4 daughter cells. The parental cell, it is diploid, but the daughter cell, they are haploid. For mitosis, both the parent cell and the daughter cells, they are diploid. And but mitosis, you will go from 1 parental cell into 2 daughter cells. So let's take a look at the meiosis 1, the major event. So we have prophase. Now we're going to talk about each one here. Okay, no, we won't talk about each one. For prophase 1, it is very similar to the mitosis in that, of course, the nuclear membrane will disappear, but in addition to that, the homologous pair, that means your paternal chromosome and your maternal chromosome, they will line up. So the paternal homolog and the maternal homolog, they will line up, pairing each other. So each pair of chromosomes, they line up. What do they do when they line up? So we call this tetrad. When the paternal homolog and the maternal homolog, they line up, we call it tetrad. Tetra meaning 4. Why? Because we have 4 chromatids. We have 4 chromatids. So 2 from the father and 2 from the mother. So why is this so important? Because they'll do something called crossover. So remember, one is from your father, one is from your mother. So paternal homolog, maternal homolog, they form a tetrad. You see that 4 chromatids line up together. During crossover, what happens is that the chromosome, they will start to rub against each other. They'll touch and rub against each other. When they're doing this, then the genetic material, the DNA from the chromosome, they will cross over. That means they will jump. They'll move to different, randomly. It is a random process. Some of it will move to the paternal one, some of it will go to the maternal one. And then after meiosis, then you have some cell that will contain the maternal gene, and some cell that will contain the paternal gene. I like to talk about this. Sorry, talk about this. I like to give you an analogy here. Give the student, it's like, try to imagine your parents, they are having sex. Yes. This is how your parents makes you. So when they are kissing, grinding, touching each other, what's the ultimate goal? The ultimate goal is to, well, exchange body fluid. But in this case, instead of exchanging body fluid, your mother chromosome and your father chromosome, they exchange genetic material here. So it is like I call it it's like, think about how your parents have sex to have you. This is what happened. And then meiosis 2, as I mentioned, meiosis 2 is basically the same as mitosis, because it is splitting of the chromatids. So we said that meiosis 2 is almost exactly like mitosis. So you have the haploid cell produced by meiosis 1, and then they undergo this, and then they will become it undergoes the meiosis 2. The haploid cell will become 4 haploid cell. The 2 haploid cells from meiosis 1 will become 4 haploid cells at the end of meiosis 2. So why do we need to have all this? How meiosis looks? How meiosis go through all these steps and produce 4 daughter cells and so on? The main reason is for genetic variation. We talk about genetic variation maybe on Wednesday. If not, then we will definitely talk about it on next Wednesday's lecture. Genetic variation is important for evolution because it will increase the chances of our offspring's survival, the survival of our offspring. So, second bullet point is that mutation is the ultimate source of genetic variation. Well, but mutation occurs, let's say if you have radiation or if you have chemical mutagen, if you have chemical, you can also call mutation. High temperature also called mutation. But if none of these occur, can we have a genetic variation? Yes, we can still have genetic variation without mutation. And that is through the normal meiosis. the process of making gametes, they will enhance the genetic variation. So alleles, we'll talk about alleles, more about alleles when we talk about genetics. So the genetic variation is one of the reasons that causes your children, they look different from you. They look different from you, but they look similar. In certain way, they look similar to you because the gene that they got, they're basically from you and your partner or your partner. But they look different because of the genetic variation caused by the meiosis. So what are the two ways meiosis causes genetic variation? The first one is crossing over. I already talked about this. That means your parents are having sex. So what happened is that during the crossover, remember which stage crossing over occurred in meiosis? So it already says in here, it's prophase 1. Remember this crossing over occurs in prophase 1 in meiosis. So because your paternal homolog and your maternal homolog, they are grinding, touching, rubbing each other, then the genetic material are transferred to each of the chromatids. And then there's the second one, we call it the crossing over. That has another name. We can call it can be called genetic recombination. We can call it genetic recombination because the gene from one DNA goes to the gene to another DNA, and vice versa. That's why we call it recombination. As you can see, that after crossover, the 4 daughter cells, one have the maternal homolog. The other one, mostly maternal homolog, but it has some of the paternal DNA. This one, mostly paternal homolog, but it has some maternal DNA. And then the last one is the paternal homolog here. So you can see that each of the daughter cells, they are different. Because of that, it increases the genetic variation. Now the second method to increase genetic variation is called independent assortment. Independent assortment occurs in metaphase 1. So remember, metaphase 1, we have the independent assortment. So what does it mean? So there are different ways of lining up the chromosome. For example, you can line up the chromosome. Okay, maternal homolog, they are on one side, and the paternal homolog, they are on the other side. But you can also arrange it into this way. The mother chromosome, father chromosome, and then this side you have father chromosome and mother chromosome. So by arranging the chromosome in different ways, then you can also increase the genetic variation. Because when the cells split here, which is the telophase 1 or metaphase 1. So the cells split into 2. This is by the telophase 1. After telophase 1, during the metaphase 2, metaphase 2 will split the chromatids. As you can see that if the cell arrange the chromosome like this, then you will have these 4 combinations. But because you can arrange the chromosome in a different way, by the end of the telophase 2, at the end of the meiosis, you can have 4 different cells. So in this case, because of the different arrangement of the chromosome during the metaphase 1. Remember, independent assortment occurs in metaphase 1. Then you can generate 4 or 8 different cells. So programmed cell death, we talk about apoptosis. Apoptosis. Well, let me see. Oh, sorry. One last thing I forgot to mention. So, to increase genetic variation, you have crossing over, independent assortment. But the last method is random fertilization. Random fertilization is the last one because, okay, you have one egg, but then you have like millions of sperms trying to fertilize this one egg. And each sperm, they have different genetic composition. The haploid in each sperm, they are different. Because of that, the fertilization, the result of the fertilization will be different. And then the next fertilization occurs, the egg, the next month the egg will be different from the egg from last month because of independence and assortment and crossing over. And then you have another million of sperm, each one will be different, fertilize a different egg. Because of, we call this a random fertilization. So programmed cell death, why do we need to have programmed cell death? Well, of course, it is to inhibit cancer or tumor. But if you have no cancer or tumor, why do we need it? This is an example. Our fingers, originally in the embryonic stage, they are webbed. They're webbed fingers. It's like frog. It's like frog. But because of programmed cell death, the connection between the fingers, the cell there, they will die, so that we will have separate fingers. So programmed cell death, these are the characteristics of the programmed cell death. I'm not going to go over it. But if I put this in the multiple choice in the exam, you can see that okay, A, B, C, D, E. It can be made as a multiple choice easily. So what happened? What happened during the Beijing Olympic? You will see that the overgrown of the algae. So what happened is that all the pollution from the groundwater or nitrogen runoff from the factory, they provide too much nutrient to the sea, to the ocean. And then the, we call it eutrophication. Eu meaning true, trophication meaning the feeding. That means it is the over-fertilization of the area. That means we give too much nutrient to the ocean, so that the algae, they will grow so much that they will grow so much that they will actually deplete oxygen in the ocean. When oxygen is depleted, then the sea animal, like fishes, they can no longer live in that area. And it is not a good thing, as you know, that they take out all the oxygen. And actually some of the algae, they can also die because of lack of oxygen as well. So that's why we have to legalize, we have to have a legislation to control the pollution from the industry. I think that's it for tonight.